The cell membrane is a complex structure composed of various components beyond the phospholipid bilayer. Membrane proteins serve various crucial functions within the cell membrane. Channel proteins form pores that allow specific molecules to pass through the membrane. Receptor proteins respond to external signals and trigger internal cellular responses. Enzyme proteins catalyze specific chemical reactions at the cell surface. Structural proteins provide support and maintain the shape of the cell. Let's look at another important component of the cell membrane: cholesterol. Cholesterol molecules are interspersed throughout the membrane, providing both stability and flexibility. These molecules help maintain membrane fluidity at different temperatures and strengthen the barrier function of the cell membrane. Next, let's examine glycoproteins and glycolipids, which are essential for cell recognition and protection. Glycoproteins are membrane proteins with attached carbohydrate chains, while glycolipids are lipids with attached carbohydrates. Together, these carbohydrate-containing molecules form the glycocalyx, a protective sugar coat on the cell surface. The glycocalyx plays crucial roles in cell recognition, cell-to-cell adhesion, and protection against mechanical and chemical damage. The specific composition of membrane components varies significantly between different cell types. For example, nerve cells have high concentrations of proteins and cholesterol to support electrical signaling. Muscle cells contain many transport proteins for calcium and other ions needed for contraction. Immune cells have abundant glycoproteins for cell recognition and communication with other cells of the immune system. Each membrane component plays a specific and crucial role in maintaining cellular integrity and function. Cell membranes maintain an electrical charge difference between the inside and outside of the cell. This difference is known as the membrane potential. The resting membrane potential is typically around -70 millivolts. This means the inside of the cell is negatively charged compared to the outside. This electrical gradient is maintained by the unequal distribution of ions. Sodium and chloride ions are more concentrated outside the cell, while potassium ions are more concentrated inside. This membrane potential is essential for nerve impulse transmission. When a neuron is stimulated, it generates an action potential, a rapid, temporary change in membrane potential. During an action potential, the membrane rapidly depolarizes, briefly becoming positive inside, then repolarizes back to the resting state, often with a hyperpolarization phase before returning to baseline. Beyond electrical signaling, the cell membrane also participates in chemical signaling through receptor proteins. Receptor proteins embedded in the membrane bind to specific molecules called ligands, such as hormones or neurotransmitters. When activated by ligand binding, receptors trigger internal signaling cascades. These cascades involve a series of protein interactions that amplify and transmit the signal throughout the cell. These signaling pathways ultimately lead to specific cellular responses, such as changes in gene expression, enzyme activation, or metabolic adjustments. This sophisticated communication system allows cells to respond to their environment and coordinate with other cells in multicellular organisms, enabling complex physiological processes like development, immune response, and neural signaling.